I'm Mariette DiCristina, President of the National Association of Science Writers, and we're going to be talking about information technology in this next session. The title of the talk is Where Computation and Fabrication Meet, Using Computers to Mimic Biology. And I'm going to read a little bio to you of our speaker, Zack Booth Simpson, who is a research fellow at the Center for Systems and Synthetic Biology at the University of Texas at Austin. Zack Booth Simpson is an artist, scientist, and entrepreneur. A high school dropout at 17, he became a top video game designer, and at 23 was named Director of Technology at the video game company Origin Electronic Arts. He later started making interactive art, and his artwork is now in permanent installations in more than 40 museums around the world. In 2003, he began working with the Center for Systems and Synthetic Biology at the University of Texas at Austin, where he works on projects ranging from chemical kinetics to synthetic biology. His entrepreneurial efforts include 2 art-based companies and a web-based bioinformatics company, and I can't wait to hear what you say. Thank you. Please come up.
Well, thanks for having me here. Okay, so this is the most complicated slide of my entire slide deck. It's just attempting to describe what I do. So, as you heard, part software engineer, artist, game developer, molecular biologist. A lot of my friends have said that for someone who claims to be all those things, I spend frequently a large amount of time laying bricks. So maybe I need to add bricklayer to that. Okay, so here's the 20-second rundown of my life. Dropped out of high school, never went to college, became a software developer, made video games like Ultima, Wing Commander, Ultima Online. Started making artwork. Most of my artwork is sort of scientifically related. About 6 years ago, I started hanging out at the at the Center for Molecular Biology here. And after a few years of doing that, they sort of made my position official, so I became a research fellow here at the Center for Systems and Synthetic Biology. And so I also have various other careers. I'm building several art companies and this Web 2.0 company, which is trying to crowdsource phenotype information. And my, quote, real job, the one that pays most of the bills, is my artwork, which is uses computers, projectors, and cameras to make full-body interactive installation art. So some of it's sort of something like this where, is this working? No. It is? Oh, you just barely see it, right, okay. Over here, something like a piece is projected onto the floor and you walk out and plants grow from your feet, to more scientific ones such as this. This one is an RNA that you can walk up and pull apart and then folds back up. And this one's a molecular simulation of salt, so you can kind of freak out in front of it, adding kinetic heat to it, and the salt crystal melts. So these are installed in a lot of science and children's museums around the world. So somebody said, so you're an artist for profit and you're a scientist for fun? I said, no, I'm an artist for fun and profit and a scientist for fun. Okay, so here's what we're actually going to talk about, which is one of my science quests. So I'm going to start with a sort of big picture challenge question here, which is, if whales and trees can be built from seeds, which after all they can, they are built this way, then why can't we build submarines and cell phone towers this way? So I take this question very seriously. I think it is possible to build things this way. But I don't think we're going to be doing it anytime soon. And I'm interested in how it is we learn to accomplish something like that. So the first question is, why would you want to do this? Why would you want to build something like a submarine from a seed? And my explanation for this is that biological forms have really unique properties that human technologies tend to lack. Obviously, they grow, they repair, they self-sense, they adapt. And it's exactly the absence of those properties that tends to make our human technology so brittle. I mean, an airplane spar does not have embedded sensors that detect the stress and detect when the fatigue is about to break the spar. And even if it did detect it, it wouldn't be able to repair it. Contrast that to a bird's wing, which has internal sensors, knows when the stress is getting too high, and if it breaks, can regrow both the joint and, both the bone and the muscle around it. Human technology throughout history, from the beginning of the Copper Age to now, we always build things top down. We impose order on systems from the outside. From the ancient times, we've done this by digging troughs in the ground, taking melted copper and pouring it into a tray and to impose the order on the copper. To modern times, we use 3D printers to design something in a computer and then print it out on a 3D printer. So here's a couple. If you've never seen 3D printers, they're pretty fun devices. Here's a small scale one. This isn't working at all, is it? No. Okay. Okay. In the middle here, we see like a toy home 3D printer, and here we see a literal home printer. Somebody's proposing building a concrete printer. Which, by the way, I'd just like to say, if you do build a concrete printer, please don't use it to build identical little Replo-like apartments like this. So in contrast to biological technology, which is always built from the bottom up, when an organism grows, it's executing a program to grow, right? A cell, a living thing, is a computer. It is in a constant process of computing. It's making measurements about its surroundings, it's making decisions about what it should do, and it sets the state. An excellent example of this is developmental biology, the biology of multicellular organisms such as ourselves growing. From a single cell, that hundreds of billions of cells end up working together, and they do this by communicating with each other and establishing, like, oh, you're going to be a liver cell, and I'm going to be a vein, and you're going to be a heart, and all this has to get worked out computationally. It's important to understand that this computation does not end. It doesn't run and we get large and it's done. The computation is continuous. It's running all the time. It's the word biologists would use as homeostasis. There's a constant calculation of what needs to be done to maintain ourselves. And you can kind of define death as the point at which this computation no longer takes place. So, from a human point of view, we tend to treat the technologies of computation and construction totally separate from each other. And we've really conquered these. Think of the computers we've built, just totally mind-blowingly awesome. Think of the progress we've made in construction, from building skyscrapers to little tiny nanoparts. But we don't really have any technologies which live at the center of those two things. That technology that's in between is the exclusive providence of living things right now. That is to say that living things, when they're performing computations, they intimately integrate with the matter around them. It's very difficult sometimes to even disentangle what are they doing for the purposes of constructing themselves versus what are they doing for the purposes of computing a result. The two concepts to a living thing of construction and computation are so intimately connected, it's hard for us to even disentangle them. Now, this idea of building things from the bottom up, if you follow the literature at all, you've heard this word nanotech before. And the thing is about nanotech is that it's often described as a problem of we're going to figure out how to place atoms in specific configurations, and we're going to build things from the bottom up. But the point I want to make here is that even if we had a fully functional nano toolkit right now, you handed me some, like the hacker's box of nanoparts, and it all worked perfectly. It did all the amazing things that is promised for nanoparts. Even if you gave me that, I would not be able to build you a whale, because we don't know how to write the programs that would construct whales. We're not even remotely close to knowing how to do that. I couldn't write a program that created a hair. I couldn't create even a part of an eye. We know absolutely nothing about how to write the programs that are being executed, similar to those programs being run by biological things. So a sort of pithy way of saying this is that nanotech is not just a hardware problem. It is a hardware problem, but it's not just a hardware problem. It's also a software problem. In software, I mean algorithmic. It's an algorithmic problem to figure out how it is that we create pattern, the kind of pattern we see in the spectacular examples of living things all over our Earth. So we're in the infancy of understanding how to make pattern in a controllable way from algorithm. So here's a couple examples over here: a Turing pattern, a simple little fractal that created a kind of fern-like thing, and a diffusion crystal. This is like state-of-the-art pattern formation, right? This is so far from a whale or a cell phone tower that it's, laughable, right? But we got to start somewhere, and that's where we're starting right now. If we could create something like that at the interface of not just simulation, but at the interface of actually constructing things with those sorts of patterns, then we'd be taking baby steps towards this future. Okay, so that's the big idea. Now we're going to start getting into a conceptual framework for how we might start taking these steps. And the framework for this I like was a term coined by Abelson and several others in 2000. They called it amorphous computing. So the idea of amorphous computing is that you have some sort of logic device, call it a computer or computational unit if you want. But think of this logic device very simple. This is not some sophisticated CPU with a 3 megabyte cache and all this. This is just some simple little logic unit. And you take this logic unit and you imagine that you can mass produce it, like mega mass produce it. So you produce a bazillion of these things and you spread them out on a surface or in space or on a line. You spread them out somehow and you apply the following rules. They're all identical, although some of them might not be functional. They're arrayed through space somehow, but not necessarily evenly. Each processor is simple, as I described. There's no shared bus, no shared clock. They don't have a priori knowledge of their position in space, and they can communicate only locally. So it's not like the internet. They don't get to send messages from one node to another through space. They only get to talk to their neighbors. So this description of an amorphous computer is really a good description of biological form. That's what the cells in our body are. Each little cell is a little pretty humble little processor. It only gets to talk to its neighbors by chemical or mechanical means. It doesn't have any a priori knowledge of its position. It's not like there's some sort of GPS unit that allows each cell to determine where it is in space. And yet, we can see from all the living things around us what's possible with this framework. Okay, so this is to give you some idea. This actually is taken from an MIT graduate student who worked on this amorphous computing idea back in 2002, so I borrowed her slides. Okay, here's the idea. Each of the little dots on the screen, this is the left side you can see is one panel, and the right side is another panel. So here at time equals 1, time equals 0 on the left, each little dot represents one of those processors, and they've just scattered them randomly on the surface. And they've marked two little areas in red, if you can sort of see them there. And these areas were sort of exogenously indicated by the creator of this system, like say, aha, you two little areas, you're special. And here's what's special about you. You're the endpoints of a line. So through magic of some more computation that I'm not going to explain, these two lines sort of start sending out gradient feelers for each other, and they find each other, and they manage to form a line. So you see on the second panel, this is after some amount of time, that a line has formed between those two endpoints. Now, because it's amorphous, it's resilient to injury. So here, they've ablated out some section. In the simulation, of course, they just deleted them. If this were a real thing, you can imagine, taking an eraser and erasing them or ablating them out with a scissors or something. And when they're broken, the thing really, really wants to form a line. So what does it end up doing? It ends up routing around the injury. So without going into a lot of detail on how this algorithm works, it satisfies the description I told you before. It's all local communication. It's all very simple processors. And this very simple little algorithm, you can tell it smells like something living. You injured it, it regrew. Right? There's no technology we have physically implemented that maybe there's a few that come close to that. But like how simple an algorithm that is, it definitely reminds you of something that's living. Okay, so here's the question I asked. I saw those simulations. I was like, wow, that's really cool. I'd like to find a way to do that more than just simulate. I want to actually make things that execute those algorithms. So I asked, can we do this biologically? So I proposed a project which took a long time to implement, but we have actually successfully implemented. And step one of this project was UT, in collaboration with our friends at USF and the Voight Lab, we engineered a system where we took the light sensing domain out of a cyanobacteria and inserted it with some clever hackery into an E. coli, which is a gut bacteria. And gut bacteria, of course, know nothing about light. They live in the total darkness. So we had to take the light sensing unit out of a cyanobacteria that cares about light and insert it into an E. coli. What we did was we basically told the bacteria, if you see the darkness, then precipitate out a sugar which leaves a black stain that's visible to the naked eye. So what you're seeing here is a field of bacteria. And we just grew these bacteria up, spread them out on a plate, and then shined an image down onto the plate and basically took a photograph. So each bacteria was, hey, I'm in the dark. Okay, I'm going to start making a sugar. And then after a few hours, we take it out and we look at it, and there's a photograph of what we projected onto it. So in this case, for fun, we projected a picture of Darwin. So that was a Nature paper in 2005, but that was really, for our plans, only step one of what we wanted to achieve. Because this isn't really much of a computation. You might say this is a degenerate computation. The entire algorithm is, if dark, reduce dark. It's not much of a program. Okay, so I had a little higher aspirations. An aspiration for this was to conduct an image processing filter. So if you've ever used Photoshop, you know, you like take up an image and you can pull down like the filters in Photoshop, and there's a filter called edge detect, and it will find the transition between light and dark. By the way, it's also a filter that our eyes do naturally. Our retinas have a built-in edge detector, which is why we're very sensitive to edges. You know, which is why when you look at cartoons, they seem very readable to us because it matches the edge detection processing that we do naturally. Okay, so here's what we did. Same idea. We're projecting an image down onto a plate, but we've engineered a new strain of bacteria now that's more sophisticated than just a camera. So we have a mask, got the light shining down to it. What we're hoping is to find is, you know, here's this black-white binary image, and we're trying to compute this edge, the edge between them. And up here we see projecting down this black square. The next one over is a simulation of what we expected it to do, and the final one on the end is the actual system. So that's bacteria computing where the edge is of that projected image. So that's an honest-to-God computation. I wouldn't call that a degenerate computation. Here's the algorithm that they're executing. Basically, they say, each bacteria says, if and only if I'm in the dark, then I'm going to send a message. I'm going to generate a little chemical and send it to my friends. If I'm in the light and I smell that message, then the only possible explanation is I must be at an edge, right? Because I'm in the dark, I smell the material, I must be at an edge. If that's the case, I get the bacteria to raise their hand. The way I get them to raise their hand is the same way I have them precipitate out a black sugar that's visible to the naked eye. So that you can sort of see the idea of each little individual bacteria represented there as a little rod, and you can see the little green dots represent the message that the guys in the light are generating, and the black ones are the ones who've raised their hand to indicate that they're at an edge. Okay, to give you some sense of how complicated this was, this is the circuit down at the bottom. It involves 1, 2, 3, 4, 5, 6, 7 genes, which had to come from all kinds of different organisms and get pulled together, none of which are characterized properly, and it involved 1, 2, 3, 4, 5 promoters. This was a gigantic engineering effort. If you were to do this electronically, it would take you about 13 seconds. But biologically, that was a 5 calendar year project. That was an entire grad student's career to build that circuit. So you may have heard some about this project or this whole new field of synthetic biology, and there's a lot of valid hype associated with what's possible with this. And this, I dare say, is one of the more sophisticated working synthetic biology projects that's ever been executed. And there's a movement called BioBricks, which is an attempt to standardize the biological parts for this in a very engineering way. And this is a very valiant effort, but I want to try to put a little clarity on the dirty, dark secret of synthetic biology. And that is that as engineering devices, cells absolutely suck. Okay, despite the valiant efforts of the BioBricks community and all the excitement, the deserved excitement around this, cells are just fiendishly difficult objects to engineer. I mean, to begin with, we don't have compilers, we don't have oscilloscopes, we don't have debuggers. You basically are just flying blind. The only reason we got that thing to work at all was because we got lucky and a light bulb burned out in the middle of the night having the exposure time, and suddenly it worked because we had been overexposing it the whole time. But because we can't debug the things, we were just making random guesses about the way this thing should work. But here's the thing, even if BioBricks worked exactly as advertised, even if every single part was well characterized, which it's not, even if the insertion yields was 100%, which it's not, even if everything worked perfectly, cells still suck. Because cells have a mind of their own. I don't mean this metaphorically, I mean literally they have a mind of their own. Cells do not want to do what you want them to do. You try to insert a whole bunch of instructions that makes them do something fancy like an edge detector, and they go, I'm going to try with the detector and without the detector, and I'm going to see which of these is better for me. And it goes, the one without the detector is better for me, and decides to do that one. And we call that process evolution. And evolution always wins. So you spend almost your entire time when trying to engineer living things. As an engineer, you're fighting with this device, trying to convince it to do what you want it to do. It's as if, like, as a programmer, every time I tried to run a test program, the computer just decides 50% of the time not to do what I asked it to do. It just runs some other program. Maybe an older version of the program I was just running, which really is confusing. So, although there is a lot of promise, this is my little caveat, like, that people don't bother to mention, is that cells are really not suitable. They're really not excellent engineering devices because they're finicky and they don't want to do what you want them to do. So we really need a more easily engineered substrate, and the obvious candidate for this is nucleic acids. Okay, so nucleic acids, DNAs and RNAs. If you know anything about them, you've probably heard some term like that they're information-rich molecules. And this is true. They are information-rich molecules. But there's nothing special about the fact that they're information-rich. Any heterogeneous polymer like a protein is an information-rich molecule. That's not what's special. What's special is that the way they interact with each other is completely predictable. You can throw all the supercomputers in the world at the problem of predicting the interaction of proteins, and you'll be lucky if you do better than a coin flip. But with DNAs and RNAs, I can, 3 seconds on a piece of paper, I can tell you what the binding energy is going to be between two DNAs or RNAs that bind with each other. So it's the fact that they have an address space, in a computer terms, I can choose a sequence of them, and then I can predict what the probability or what the binding strength is going to be between those. Furthermore, from just an engineering practical standpoint, synthesis, sequencing, cut, copy, and paste are all implemented. So I don't have to go reinvent the most basic tools. That you might call that the soldering iron of the technology. And further, there's just the mundane but important. They're non-toxic, cheap, and readily available. Plus, you can make nucleic acids do really fun tricks. Here's the work of my friend Paul Rothman at Caltech. So what he's done is, he calls this DNA origami. He takes a long DNA strand and a whole bunch of small DNA strands, and he carefully has chosen the sequences of these strands, throws them into a soup, mixes them up, and waits. And what happens is that the little strands start binding to the big strand in a very specific way, and it starts folding up, and he's programmed that fold to make a smiley face. So down here, this is an actual atomic force microscope image of a 100 nanometer smiley face. These were assembled at 90% yield with 100 billion of them in the drop of a pipette. So as his friend Eric pointed out, that makes this the highest density of happiness that's ever been made on Earth. So speaking of Eric, whose lab Paul worked in, this is also Caltech. So Eric's taken these ideas of DNA, and instead of building things with them, he's showing what kinds of computation you can do with them. So he and his grad students have built 3 flavors of computation, actually probably even more now. And I'm just going to touch on them, and then I'm going to focus a whole lot on one of them. So crystalline cellular automata. So that's this Sierpinski triangle here. He took Rothman's DNA origami. He made a tile. It's called a Wang tile. Think of it like a puzzle piece. And these puzzle pieces come together in some very specific programmed way. And without going into a whole bunch of stuff, if you have a Wang tile, you can prove it's Turing complete, and you can do any computation with it. And what they managed to do was they managed to build a Sierpinski triangle. And actually, they've done more sophisticated stuff since then. But this is a crystalline computer. Okay, they've also done transcription-based circuits, which is what I'm going to go into in a lot more detail, because our work is based on their work here. We've modified it a little bit, so I'm going to skip over this and show you more detail when we get to the way we're doing it. Okay, we're going to pause for just a second here. I'm trying to build an information technology, and I'm trying to build it out of wetware, whether it's biological or sort of in vitro components from biological. It's wet. So you might be surprised, but there's actually very little talk between these two communities. communities. The information technology community has 100 years of experience about building devices, but they're all electrical. And so it's all in the EE department. And the chemistry and biological department, like literally on our campus, is across the street, and not a single professor like ever walks between those buildings. So with the help of my friends, I've had to like kind of try to build a bridge between these worlds. So, and originally I had a whole bunch of slides trying to explain this, and my friends wisely advised me to cut this down to one slide. So, okay, here, if you don't follow this, well, that's my friend's fault because I didn't go into enough detail. Okay, here's the idea. The basic building block of any information processing system, no matter how you choose to implement it, whether it's electrical or chemical or out of Tinker Toys, fundamentally you got to have an amplifier. So the like, you know, first sentence of a Wikipedia like introduction to what an amplifier is goes something like, an amplifier takes a small input signal and makes a big output signal with it. But this loses a lot of what exactly goes on inside of an amplifier that's relevant to understanding the mapping between an electrical and a chemical system. What an amplifier does is it sculpts a source of free energy. So it has some big supply of free energy, and it takes the input, and it looks at the input, and it sculpts the free energy into the shape of the output. Right? It doesn't transform the input. The input energy does not become the output energy. It sculpts the free energy into the shape of the input energy. Okay, that's the source. That's the sort of line I drew in orange there. Now, when the input goes high, this sculptor like takes a bunch of energy out of the free energy supply and sculpts the output high, but at some point, the output's got to go low again. If the input goes low, the output's got to go low. So that means this sculptor free energy has to have a way of like getting rid of the energy that's in there. It's got to discard it somewhere. So it has to have some path to dump that energy once it's used it. And that is some sort of sink. Okay, so I'm trying to compare and contrast like an electrical and chemical system here. When you choose to encode a signal, like we're doing with our inputs and outputs here, in an electrical system, you encode it by voltage per unit capacitance. Chemically, what you're going to do is you're going to do it with concentration per unit capacitance. So you're going to think of electrical voltage per unit capacitance, think of that as a pile of electrons sitting on top of a capacitor. It's a high concentration of electrons. Chemically, this is a high concentration of some reagent that we declare to be our signal, and a high concentration of it allows us to encode signal. Okay, the free energy in the case of electrical, say on a computer, that orange free energy path is going to come from somewhere, like the wall, the power supply that's connected to this, which is ultimately connected to some big carbon-eating machine downtown. And when we're going to do it chemically, we're also going to need a source of free energy. So if we're going to do it biologically, that's most likely candidate is going to be ATP, the universal sort of currency of power in biological systems. And finally, this entropic sink. And when you have an electrical system, you're going to dump the energy that you don't want anymore in the output, and you're going to dump it out to ground. And in doing so, you're going to ohmically heat this wire, and it's going to convert to heat, and you're going to have to get rid of that heat somehow. So on a computer, you put a heat sink there, and you're going to dissipate it out to air. If you don't have a path to getting rid of this, then you're going to pollute the system with entropy, and quickly it's not going to work anymore. So when you're designing this, you have to think about this whole path. And I kind of go into this because if you read the literature of the excitement over building circuits biologically, this is usually skipped over. And it's like, look, we made a NOT gate that worked once because we didn't think about how it was that we got the entropy out of this system. Okay, so hopefully that's a little helpful introduction to this much more complicated slide that's about to happen. Okay. So this is comparing an electrical implementation of an amplifier to a chemical implementation of an amplifier. And up here I just have the same diagram from the previous slide for reference. Okay, here's how it works electrically. Here's our input line, and we have a transistor, and connected to the top of the transistor here, that line represents a power supply. So we can think of some infinite source of energy that's available and switchable through this transistor. So when the input is low, this transistor doesn't conduct very well, so not much can go through. And when the input goes high, current can flow through that transistor and charge up that output capacitor. So what happens, the transistor starts conducting, let's imagine that output capacitor is low, and now fills the capacitor until the capacitor and the power supply are at equilibrium. Now, to get the output back down again, when this stops conducting, we got to get the power energy out of the output. So the way we do that is we put a resistor here to ground, and that gives a path for the energy to get out of this and out to ground. And it's the dissipation through that resistor where you're going to get heat going off of this thing that you're going to have to get rid of somehow. Okay, chemically, the picture is a little more complicated, but it's analogous. Okay, brief introduction. Hey, high tech. Okay, now I feel like a rock star though, until I trip over something. Okay, DNA. Let's see. The weird thing about this is I talk with my hands, and now I'm like gonna be. Okay. Okay, DNA. We have to have a brief introduction to DNA and polymerases here. Okay, you've all seen pictures of DNA. It's a double helix. There's a machine, a nanomachine called a polymerase. It's a very amazing machine. And what it does is it comes along and grabs onto a double helix and goes zips along it. And while it zips along it, it makes an RNA copy of the information that's in that DNA. How does the polymerase decide where to start and begin copying? Well, it does this because there's a special start sequence. Who cares what it is, ATGAC, whatever. There's some special sequence that some given polymerase recognizes. It goes, aha, and it grabs that special sequence and it begins a copy. So what the Winfrey lab experimented with successfully was that little region where the polymerase binds is called the promoter. And the standard way of illustrating that is to put this little arrow here. This arrow represents, I'm a place where a polymerase can come bind and start copying. So these circles represent the hydrogen bonds, the base pairing of the DNA. And this is a little different than the way Eric's lab did it, but basically we use one piece of DNA that wraps around onto itself. And notice there's a nick right here. So if this strand pops open, then this promoter will not be complete. And polymerases don't like incomplete promoters, so they won't use them. So if you can figure out a way to pop open this promoter, then you stop the transcription. So this is closed, polymerase comes along, transcription starts making output RNAs. But if you can pop this open, polymerase comes along and goes, I don't like that, and it stops making output RNAs. So this is starting to feel a little bit like that transistor over here. Okay, so what you do is, we get to choose the sequence of this loop. So we can choose the sequence so that it is the antisense of some sequence of an input RNA. So here's an input RNA, and it's wiggling around, and it finds this, and it can bind to it. And because of the interesting property of double-stranded nucleic acids have higher tensile strength than single-stranded, so they spring open. So when this binds, here it is bound in, this goes spring, it opens. So we can switch whether the output is running or not by attacking it with this strand. Now we have to be able to get that strand off, or this is a one-shot only device. If we want to reset, we got to figure out how to get this strand back off again. The way we do that is, you notice there's a little extra hanging off here. And this extra part's called the toe hold, and we designed it so that another strand can come along, bind there, and then it can diffuse linearly along here, and it's thermodynamically better for that double-stranded piece to come off, causing this to close back up again. Okay, I know that's complicated. The basic part here is that the free energy, just like before, is coming in the form of ATP, and it's going through the polymerase, which is acting to make an output RNA. And that only happens when the input is low. When the input is high, then it springs open the gate, and this gate stops making stuff, and then we have a nuclease down here. A nuclease is a machine that gobbles up any RNA it finds. Yum, yum, yum. It just chews it up into pieces. So that's the equivalent of that resistor, which is constantly pulling the output down. Okay, it's really complicated. Did everyone follow it? Okay. Okay, so the point is we're building an amplifier. An amplifier is the building block on which everything informatic we're going to do is based. Without an amplifier, you can't do serial processing of logic because your signal would die off. If if I give them, it's like the game of, what's that game called? Telephone, where you tell somebody something and then they say something and it dies off at every stage. If you don't have an amplifier, that's what's going to happen. So whether you're doing this chemically or electrically, you have to have this idea of an amplifier. And again, the reason I'm kind of going into so much detail in it is because many of the models you see for digital biological implementations fail to consider the amplifier part of it. And so they sort of do one-off tricks, and then they're like, and then somehow that will scale. Well, no, it won't. Like, you're going to have to amplify it. Okay, so without going into too much more technical detail here, basically, as an engineer, I get to choose what the sequences are for the inputs and outputs. And I can choose these sequences so that I get different transfer functions. So the transfer function is just saying how much output is there for a given input. Okay, so the way you read this is I minus means there's a lot of the anti-sense strand of the input, and I plus means there's a lot of the sense strand. So see, a variable is determined by where you are here. If there's a lot of the sense strand of something relative to the anti-sense, then you're a positive number. If there's a lot of the anti-sense relative to the sense, then you're a negative number. And if there's equal amounts of the two, you're 0. Okay, this technically is called the log ratio. It doesn't really matter. The point is, when the input is somewhere here, this graph tells you what you're going to do in the output. So with these two transfer functions, we can build an inverter. So in computer technology, there's two things you need to build a computer: a NOT gate and an AND gate. So this is a NOT gate. When the input is low, I'm going to get a high signal, and when the input is high, I'm going to get a little low signal. Now, you're just going to have to trust me. With my scheme, it's also possible to build an AND gate, but you thought the last one was complicated. It'll be really complicated if you see that one. So we're just going to gloss over that. Okay, despite the fact that I'm not going to show you any AND gates, I want to demonstrate to you that you can actually have a lot of fun with just plain old inverters. Okay, here's the fun. Now, we've finally covered a lot of ground, and we finally gotten to where the whole point of this, which is to do an amorphous computation. Remember, the amorphous computer is a computer where you've got an identical processor spread out over space somehow. Okay, so here's the processor. It's incredibly ridiculously simple. It's two inverters pointed at each other. So we've got this little unit. Think of it as a little black box with two inverters inside of it. And we take that black box and we smear it out through space. Okay. Dual inverters like this are stable. They're a latch. Basically, if you start this so that the left and right signals are very close to each other, then if one's a little bit higher than the other, then one side will begin to suppress the other side, which will let this side gain more, which will suppress this side more, which will gain this, and it'll go that way. If it starts a little in the other direction, it goes that way. So what that system is it's a coin flipping machine. If you start it right in the middle, it will choose randomly to go one direction or the other. Okay? So now here's the idea. We smear this coin flipping machine through space. And that's this diagram. Now, practically, you'd probably smear it onto a surface. But for the purposes of illustration, it's easier to smear it in one dimension and then give you a space-time plot instead of some sort of movie animation of what happens, right? Okay, so here at the bottom is time 0. And although the graph's a little bit, it's just a screw up when I pasted it in, it starts off green. Okay, red represents the system flopping to one state, and blue represents it flopping to the other state, and green represents halfway between. So think of it as heads and tails if you want. But these are just states of concentration of two chemicals. Okay, so we started off at 0 with a little tiny bit of noise, and so each little volume of space, it commits to the coin flip, and it goes, I'm either flipping heads or I'm flipping tails. Okay, in this case, there's no diffusion. So each little volume of space just goes, I'm heads, and the next little volume goes space. I'm tails, this one goes, I'm heads. They have nothing to do with each other, so what you get is the binomial distribution. Sometimes, by chance, you'll see a couple of reds in a row happen to be flipped, and then, a couple blues. The distribution of the sizes of the features in that are just pure chance of the coin toss. Okay, now it gets interesting. You take this exact same algorithm and you add diffusion. Okay, so now what happens is each time the coin flip takes place, each little volume of space is to recruit its neighbor. So let's say this volume of space starts to go in the heads direction, and it starts emitting head signal to its neighbors. So if its neighbors are like kind of not sure whether they want to go heads or tails, they might change their mind because they're getting swamped by head signal. So especially if there's a great big area of space like this, they're going to fight a really good fight because they're all working together, sending out blue signal to their neighbors. So they're going to end up recruiting more of their neighbors to the blue side. So that's the result, same starting condition over there. Here you can see exactly, as time evolved this way, this probably was going to go blue, but it was surrounded by red, and red overwhelmed it and converted it to red. So the result is that you get much larger feature sizes with diffusion. Okay, that's a great question. The question is, what are we physically doing here? Okay, so these, of course, are simulations, but when we do it for real, it will be done in it will be done in a gel, spreading the DNA into a gel and then watching it evolve over time. And it's as if I'm plotting, what the gel looked like at time 0, what it looked like at time 1, et cetera. So we're looking at the evolution of that one-dimensional gel through time. But nobody says you have to do it one dimension. You could do it anything. It's just easier to plot this way. Yeah, so the question is where is this on the digital analog spectrum? And the answer is that's a really good question. That's a really cool thought. Okay, so it gets a little more interesting. Okay, so first I want to make sure everyone understood what happened. The picture on the right is a picture with high diffusion relative to the reaction rate. So the result was that each little area of space was playing a recruitment game, and they're battling it out with each other, and they end up forming some feature set. Okay, that was with 2 inverters. Now we're going to go to 3 inverters. So this is still an incredibly simple circuit. When you have an odd number of inverters, it's unstable. It's called a ring oscillator. Same game as before. We started up randomly. And so each little volume of space down here, it's like, okay, it's sitting there, and it starts to say, okay, it begins oscillating and finally gets fully oscillating. But because it was started randomly, it has no particular preference for the phase at which it begins. So each of these volumes of space is just randomly oriented in phase. They have nothing to do with each other. Okay? Now, just like before, this is now the simulation with diffusion. So now, again, each little volume of space tries to recruit its neighbors. So when it's at some phase, it's like, hey, neighbor, I think you should come over to my phase. And they're all working together trying to recruit each other. And so these, again, are exactly the same starting condition. And what happens is, by chance, you'll happen to get some area that was sort of by chance phase aligned. So these guys are all working together now trying to convince their neighbors to move into their phase. And as you can see, they start to spontaneously synchronize with each other. So if you continued the time simulation up a couple more, you'd see just perfectly horizontal lines. They'd become completely phase locked with each other. Okay, that idea of spontaneous synchronizing phase locking is a very old idea. It was first described by Huygens in the 17th century. He noticed it while he was sick in bed, and he had some pendulum clocks. Apparently they were in this configuration because this is the drawing he actually made. And he noticed that these clocks spontaneously synchronized with each other because they were transmitting information through the vibration of this board they were hanging on. So as the pendulum swung, they were causing this little resonance, and that little resonance ended up, the lowest energy form was for them to be either synchronized or anti-synchronized. So spontaneous phase alignment is a very famous sort of the building block of emergent phenomena. So if you read about complexity theory and chaos theory and all that kind of stuff, there's entire very good books about this idea of spontaneous synchronization. And all begin with Huygens' observation in 1665. So what we just saw there was a simulation that we think we can do in DNA to sort of recapitulate that. Now, why that's so cool is this. This is exactly the same thing as I just showed you. It's an amorphous oscillator. The only difference here is I did it in a bigger space, and I hand-picked out one for you. They don't always completely completely synchronized. The reason is because what happens is there'll be by chance an area that picks some phase, and they battle it out with each other and they come to an agreement to pick a phase. And meanwhile, the same thing happens over here. And by chance, it'll happen that these two areas are 180 degrees out of phase with each other, and then there's no consensus at the border about which phase, and they fight over territory. And that's what you're seeing happening here. Here's a region that has decided on a phase, and here's a region that's decided on a phase, but the boundary between them is 180 degrees off, and they're in a war trying to establish who's going to win, and you see this phase boundary defect propagate through space. So my point here is that this is just an oscillator plus noise equals pattern. You can get stuff that looks vaguely biological by doing the dumbest thing in the world. Okay, everything I've shown you so far is a scale-free simulation. So we're in the process of building these things, so of course we're going to go and try to do an estimate of what the actual scales are here. And so, using the best numbers we could get out of the literature, our estimation came out to be in tens of hours and tenths of centimeters. So why is this important? Remember, these are molecules that are executing this. These are nanometer devices. The emergent property of this nanometer computation is centimeters in scale. So this is honest to God like nanotech emergent, right? It's a tiny device emergently creating structure at the macro scale, right? Now, this hours, tenths of centimeters feels in the ballpark, at least within a couple powers of 10, right? Because if you think of something like a plant, if you had a plant making pattern at the centimeter scale, you'd expect that to take days to weeks for it to grow that. So the fact that a smaller system can make pattern in tenths of centimeters in hours, that feels about right to me, as compared to, I don't know, seconds and micrometers. So these systems are going to grow slowly. Don't expect these things to out pops a whale, right? I mean, these things are going to have to grow very, very, very slowly in the same way that we expect biological things to grow. Okay. So we're currently building those two systems, the ring oscillator and the latch, and we're trying to build those out of DNA. Hopefully my grad students are doing this literally as we speak, but they're probably not. And our ultimate goal, we haven't actually started executing this, but this is our goal, is to create a stadium wave. So you all know the stadium wave, like, whoa. Okay, so what I want to do is create the amorphous DNA chemical version of the stadium wave. So there's going to be two variables. One variable is the standing variable. When the state of this variable is low, you're sitting. When it's high, it represents standing. And when the state of this other variable called tired is low, well, you're excitable. And when it's high, you're tired. Oh man, I just want to sit back down again. Okay, so think of how a stadium wave works. You have everybody sitting, so everybody is not standing, not tired. Okay, somebody comes along and somehow exogenously stimulates this. The cheerleader's like, ah! Okay, so some little region in the stadium stands. And now the people next to them see them standing, they're like, oh, okay, I'm excitable, so I'm going to stand too. And the person next to them says, I'm excitable, and I'm going to stand. And everybody stands for a brief moment until you stand for a while, and you start accumulating tired and getting more tired, and I think I'm going to sit now. And you sit back down, and you're like, yeah, boy, that was kind of fun, but I'm not going to stand back up. If somebody else tries to get me to stand, it's going to take a while before I'm willing to do it again. And that behavior creates a propagating wave. Okay, so I'm not going to go through all the details of how this circuit works, but suffice to say it takes only 3 gates and two constitutive motors to make this happen, which is really kind of surprising. It's really a simple circuit. There's no ands in it. It's nothing but amplifiers and inverters. Okay, so here's the simulation of it. Again, a space-time plot. Here's time, everybody's sitting down, and at this point right here, I exogenously added a little bit of the standing variable. So it's as if my pipette was a cheerleader and got this little group to stand. Okay, so as time evolves this direction, right, people start standing on either side of it. The wave starts traveling in both directions, and then the guys who were the first one to stand are get tired and they sit back down again. And so now you have 2 pulses traveling out. So for the purpose of illustration, think of this as a circular stadium that I snipped and unfurled, right? So this edge is the same as that edge. So the pulse starts, the wave goes whoo around the stadium and collapses on the other end. And it does not continue. Why? Because there's tired people on either side. So it goes around and just cancels itself out. Okay, that was a symmetric starting condition. Now we're going to do a simple asymmetric starting condition on the other side. Now I pipette in a little bit of standing, and right next to it I pipe in a little bit of tired. So now the standing guys start to try to propagate, but they can't propagate in this direction because those guys are tired. So they propagate only in this direction. So it goes standing, standing, wave, 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 all the way around the stadium, all the way back around the stadium to where it began. And by that point, the tired guys have recovered, and they'll just continue to propagate the stand. So now we have a wave that's just going around the stadium, propagating around the stadium, right? Okay, so that is the system that I want to build out of a DNA amorphous computer, and that's probably years away, but that's what we're trying to build. Now the thing about this is that there's only 5 parameters of this system, actually 6 if you count this resistor. It's the concentration of these 5 gates and the concentration of this resistor. You have to choose those 5 parameters right. If you don't choose them right, it's not going to behave like this. So you know, this whole engineering game of trying to find just the right set of differential equations is going to make this happen. But if you just go and, like I did, naively build a simulation of this and you have no idea what those parameters are supposed to be because you're just stupid enough like me to just try it and see what the hell happens, then what you get is crazy stuff like this. And so you go, that's a bug. It's not doing what I want. Or you go, holy crap, that's cooler than I wanted. That's even cooler than what I was trying to build in the first place. It turns out that this system is more rich with this kind of stuff than it is with that stable linear wave. And if you've ever read things like Wolfram's, New Kind of Science and all that kind of stuff, you immediately recognize this as a 1D cellular automata. So that incredibly simple system, 3 gates, 2 promoters, creates stuff that looks shockingly like biological systems, as has been noted by lots of people who've worked on digital cellular automata, that things like shell patterns look just like this. And so if we can make this work, what I'm trying to do is get that built out of DNAs. And ultimately, if I can convince the DNAs to make this state, then the output of that can be a Rothman pattern. So think wherever it's red here, the output is I could tell the system to go do something like that. So it would think where red, make smiley face, where not red, don't make smiley face, or much better, where red, make a DNA strand that kind of glues amorphously together. So you've got a strand with another strand with another strand with another strand, and that's called a gel. And where you're blue, don't do that. And so what would you do? You'd make a pattern gel. So imagine that thing, which is a physical gel, which means it has filtration properties, right? So if this thing's a physical gel, and let's imagine it's on the centimeter scale, then I could pass a material through this thing, and there's now this is a filter, right? Some small things are going to get through, and some big things are going to get through. And if I change the parameters slightly, a lot of bigger things get through, right? So this is exactly the kind of game that evolution uses on real things. So imagine that this gel was a sponge, and a sponge is making decisions just like this. It's like, how big should here I am in the ocean, and like, how big should the pores in me be to optimize the filtration through me for feeding? And it's probably a little bit different if you're at one depth or at a different temperature. And so evolution would dial this parameter. What would it be dialing? It'd be dialing the concentration of those gates in order to build a phenotype that had certain mechanical properties. And so, okay. So here's the summary of this. Life is at the intersection of computation and construction. And we know next to nothing about how to build devices that live at that intersection, but we look around us and we see all the amazing things that life does, and it's just obvious that we should try to learn those things, sort of replicate those things and take advantage of those algorithms. So we're trying to take little baby steps about learning how to build lifelike amorphous computers doing this. So this entire talk is basically an engineering talk. So this is a group of science writers, not a group of engineer writers, so you might be questioning, well, what's the science in this? And here's my answer to that. So biology for hundreds and hundreds of years has been a deconstructive science. All we can do with biology is we can look at it, take it apart, and try to describe it. We've never been able to forward engineer it. And without a discipline of forward engineering something, it's very, very difficult to make progress in science. And this is most concisely stated by Feynman in his last Blackboard. He had written, "What I cannot create, I do not understand." Without the ability to create simplistic, lifelike devices, we really can't understand biology. Another analogy might be, as long as we were trying to mimic birds, we never succeeded. It was when we gave up trying to mimic them and started an engineering discipline of building wind tunnels and isolating lift from drag that we began to make progress. And only once we had the engineering discipline of aeronautics were we able to go back and ask the questions, how do birds fly? So engineering and science are coupled to each other. You have to have both of them to make progress. And biology has been constrained for centuries by the fact that it has not had an engineering discipline that goes along with it. And we're in the infancy of changing that. With synthetic biology and like nucleic acid amorphous computing like this, we're now going to start an engineering discipline that will inform us of how to ask questions about living things. By the way, I knew this quote for a long time, so writing this talk of did what anyone would do, I Googled that quote to try to go find the actual Blackboard, and I didn't know what else was written on Feynman's last Blackboard. Something only Feynman could have written. Know how to solve every problem that has ever been solved. And over here he's written, to learn, and apparently there's only 5 things left on his list. And he's already knocked one of them out in the morning before he died. And ironically, the last one on the list says nonlinear chemical something or other, and I take that to mean nonlinear chemical systems, which is what this whole talk was just about. So I guess I'm just trying to, he was going to, if he had just lived a few more hours, I guess he would have figured this all out. So the summary of this is learning to write programming, what I'm talking about, is to understand life. I really strongly believe that we will not understand all the intricate aspects of living things until we have a forward engineering discipline that tries to mimic their properties. So thanks for having me. Thanks to John, who's here, and Andy and Edward, Ji Chen, Nam Nguyen, and all the College of Natural Sciences for letting a high school dropout like me come play with their toys. So thanks.
Thank you. That was great. I think we can take 1 or 2 questions if anyone would like. Please come up to the mic.
Well, that was dazzling, and it may be because I'm dazzled that my question might be naive, but if you're aiming for certain design goals and you are able to translate them from simulation to bioengineering, can't you then let evolution lend a hand and throw a whole bunch of variations out there and see which ones come closest to your goals?
Okay, so that's a great question. Like, to what degree can we use evolution? So there's a whole nother hour-long talk associated about that. And but the quick answer to it is related to the one slide where I said cells suck. So here's the thing. On the one hand, it's a great strategy, and we use strategies of evolution to design molecular systems. But there's a real problem with this, and as you make your visions grander and grander of evolving these kinds of things, you end up in the same problems of cells suck. Devices that evolve rapidly will quickly evolve to not do what you want them to do. They want to solve their immediate phenotype problem, which is living inside of the little, petri dish that you have them. They don't necessarily want to solve the problem you want to. So selection engineering, like you're talking about, is an emerging field that will contribute to not just this system, but many systems for, all the future, but is a whole nother can of worms of selection engineering, which is, does that answer the question? Okay.
Okay, last question.
Okay.
Yeah, sorry about that. You emphasize in your amorphous computing about local interactions, and of course that's been a big theme in cellular automata and complex systems in general. But in real organisms, particularly multicellular organisms, there is long-scale communications, endocrine, hormones, all the rest, and that's actually very important for overall coherence.
Yep. So. Oh, absolutely. So, look, I'd be happy if I could build something that even remotely looked like a sponge, never mind like something that was there's no doubt that long-range communication takes place in complicated organisms like us or whales, but it's like I can barely even think about this problem yet before I start adding that to the equation. But you're absolutely right. That will become a part of any quote-unquote real lifelike system.
Right. Thanks.
Thanks so much. I hope that Zack Booth Simpson will be available for a couple minutes after. I'm sorry I had to cut the question short a bit, but to